Hello students, coming to next class, endomembrane system. In the last class we discussed about a group of cell organelles, means here cell wall. We discussed about the plant cell wall, right, and the plasma membrane and the functions of the plasma membrane we discussed. And next one is the cytoplasm, right? We discussed about that cytoplasm and the characteristic features of the cytoplasm and the protoplasm also, right? So coming to this class, endomembrane system. Endomembrane system, a group of membrane contained cell organelles, especially here all these are single membrane contained cell organelles. Single membrane contained cell organelles. These organelles which are collectively involving a group of functions. A group of functions. A group of cell organelles are collectively performs a group of functions. Especially that function is nothing but protein. We know about that protein synthesis, and we know about that activation of proteins. We'll discuss now. We'll discuss about that protein synthesis process and activation of proteins, right? And the transportation of proteins. Basically, what I'm telling here, a group of cell organelles are involving synthesis of proteins, activation of proteins, and transportation of proteins. Not only proteins, along with the proteins, some of the other molecules also synthesized, which are also transported. So here, today we are discussing about that endomembrane system. Within endomembrane system, there is an endoplasmic reticulum is present, Golgi complex is present, vacuoles are lysosomes. Vacuoles are seen in plant cells. Lysosomes are seen in animal cells. So that is the difference. For suppose this is the plant cell, in that case vacuoles are present. For suppose this is the animal cell, in that case lysosomes are present. Right? So that is about that endomembrane system. Within endomembrane system, there is an endoplasmic reticulum is there, Golgi complex is there, vacuoles are lysosomes are present. See, I drawn here one of the nucleus structure. Nucleus is not related to the endomembrane system. See, this is one of the nucleus. This is one of the nucleus. Within that nucleus, there is a genetic material is there. That one is nothing but DNA. That DNA, okay, this is the DNA, right? And it transfers the characters from one generation to the another generation, right? So this is about that endomembrane system. Within that endoplasmic reticulum, the very first part is the endoplasmic reticulum. That one, below that endoplasmic reticulum, there is a Golgi complex is there, and below the Golgi complex, vacuoles or lysosomes are there. So these are the three parts are there. Okay. Especially here, vacuoles are seen in only plant cells. Lysosomes are only seen in animal cells. That's the reason why I said here three, right? Endoplasmic reticulum, Golgi complex, within plant cells, vacuoles, within animal cells, lysosomes, right? So these three parts are especially seen in plant cells or animal cells, and which are included in endomembrane system. These three are included in the endomembrane system, right? So that is about that initial introduction regarding to the endomembrane system. And before going to the topic, I like to discuss about one of the important point here. For suppose, if I ask the question, cell have the life, right? And stone is there. Stone not have the life. Why cell have the life? Why stone not have the life? Your question is very simple. Cell, okay, we know about that cell. For suppose any cell we are taken, either plant cell or animal cell or human cell, any cell. Cell have the life. If you are taken any stone, or for suppose this is a pen or marker, right? Okay, this will not have the life. So what I am telling here, stone is taken here, one of the example, stone not have the life. Why cell have the life? Why stone not have the life? This is a very basic question. Here cells have the life and stones not have the life. Why here life is maintained? Why here life not maintained? This is the basic question. Because in the last class also we discussed there is in metabolic reactions that takes place. Within that cells, metabolic reactions are takes place. Here metabolic reactions are nothing but a group of biochemical reactions that takes place. A group of biochemical reactions that takes place in presence of enzymes. enzymes. So very simple, whatever that living cells are there, within that living cells, a group of proteins are act like an enzymes, and these enzymes are involving several biochemical reactions which are nothing but metabolic reactions, which are nothing but the metabolic reactions, which are maintains the life, So the metabolic reactions are maintains the life. Metabolic reactions are done by enzymes, right? So here enzymes are maintains the life. So what I am telling here, a group of enzymes which are involved in biochemical reactions, that reactions are maintains the life, right? Sir, how? Okay, we well known, for suppose one of the cell is there, DNA is very essential. How DNA maintains the life? Especially DNA is the biomolecule, that DNA responsible to maintain the life. How DNA have the capacity to maintain the life? How DNA have the capacity to maintain the life? So anybody think about this one? How the DNA have the capacity to maintain the life? How the DNA have the capacity to maintain the life? So here my answer is, for suppose the simple explanation is, there is a cell contains that nucleus, within that nucleus there is a DNA is there, right? This is one of the DNA molecule. Always DNA is the double helical structure. See, this is the DNA. That DNA synthesizes DNA. So already DNA is there. That DNA involved in the DNA synthesis process. This is called as DNA replication. Right? This is the DNA, and this is also DNA. One DNA molecule synthesizes the two DNA molecules, because during DNA replication, one copy produces another copy. Again, the DNA involving synthesis of RNA. For suppose this is the RNA, right? From DNA, RNA is synthesized. So this step is called as transcription. So synthesis of mRNA or RNA from DNA. This step is called as transcription, right? So next one is that from the RNA, this is one of the RNA, and that RNA again involving synthesis of proteins. This step is called as translation. So what I am telling here, within any cell, DNA is the main component. That DNA involving DNA replication process, synthesis of DNA from DNA, that is called as DNA replication process. Again, DNA involving synthesis of RNA. Again, that RNA especially binded with a group of ribosomes. Ribosomes are involving protein synthesis process. Here proteins are synthesized. So I said here a group of enzymes are involved in biochemical reactions. These are nothing but proteins. So here proteins are there. These are the proteins. Okay, and these are inactive proteins. Mostly majority of the proteins are inactive proteins. The inactive proteins are converted into active proteins. So this step is called as post-translational modifications. Post-translational modifications. So what I am telling here, within that nucleus, DNA is there. That DNA, sometimes DNA replication takes place, and that DNA responsible for the RNA synthesis. Within that nucleus, RNA synthesis takes place. That RNA transported from inside the nucleus to the endoplasmic reticulum. Within endoplasmic reticulum, there is a ribosomes are there. That ribosomes are involved in protein synthesis. Again here, protein molecules are synthesized. within endoplasmic reticulum, presence of ribosomes, proteins are synthesized. This is one of the inactive protein. This is one of the inactive protein. That inactive protein transported into Golgi complex. That inactive protein transported into Golgi complex. That inactive protein inside the Golgi complex activated. That one is converted into the active protein. That one is converted into the active protein. So whatever that proteins are there, inactive proteins are there, the inactive proteins are transported into the inside the Golgi complex. Some of the post-translational modifications are takes place. The inactive proteins are converted into active proteins. The inactive proteins are converted into the active proteins. That active proteins are transported. That active proteins are transported. So this is the one of that endomembrane system. Here within that endomembrane system, nucleus is not included. Nucleus is not included. Whatever that endoplasmic reticulum is there, that endoplasmic reticulum involving the protein synthesis process, that proteins are inactive proteins. Again, these proteins are activated. These proteins are activated within Golgi complex. These proteins are activated. Okay, within Golgi complex. That activated proteins are transported by the lysosomes or vacuoles. So, sir, why you are discussing about this one? Why you are discussing about this one? Okay, initially I asked one of the questions here, how the life was maintained. I said the answer is by the biochemical reactions. How the biochemical reactions are maintained? By the enzymes. By the enzymes. DNA is responsible to maintain the life, right? So how the DNA maintains the life? How the DNA maintains the life? Okay, answer is very simple. DNA responsible for the RNA synthesis. RNA responsible for protein synthesis. These are initially inactive proteins. The inactive proteins are modified into the active proteins. Once the active proteins are there, these are nothing but, the active proteins are nothing but enzymes. So I said enzymes are involved in biochemical reactions. Enzymes are involved in biochemical reactions. Enzymes done metabolic reactions. Enzymes done metabolic reactions. So presence of enzymes, all biochemical reactions that takes place. Then life is maintained. Then life is maintained. So what I asked that the first question is, why life maintained within the cells, why life not maintained within the stone? Within the stone. Because of biochemical reactions. Because of biochemical reactions. Biochemical reactions are done by enzymes. So here a group of cell organelles are there. These are collectively performs a group of functions. A group of functions. Okay. And finally these are produces active proteins. The active proteins are nothing but enzymes. The active proteins are nothing but enzymes. Enzymes are responsible for biochemical reactions. So biochemical reactions are takes place, life is maintained. Biochemical reactions are not takes place, the life is not maintained. The life is not maintained. So here the DNA is responsible for all the functions within the cell. DNA is responsible for all the functions which are occurs within the cell. So DNA is responsible to maintain all the functions within the cell. DNA is responsible because DNA synthesizes the RNA. RNA synthesizes the protein. Some proteins are involved in some types of functions. Okay, some proteins are involved in another type of functions. For suppose we are eating food. Okay, starch is there. The starch is converted into glucose. Glucose is transported. Glucose is entered into the inside the cell. Glucose is converted into energy. So a group of enzymes are required. A group of enzymes are required. So what I am telling here, see several functions. For suppose I am speaking, okay, that one is also requires energy. And for suppose we are thinking, nerve impulse transport, that one also requires energy. So every function within every organism done by biochemical reactions. Every function within every living organism done by biochemical reactions. So biochemical reactions are maintained by the enzymes. Biochemical reactions are maintained by the enzymes. So here what I am telling, endomembrane system is very important for the synthesis of active proteins. For the synthesis of active proteins. So this is just the parts which are involved in functions. Again we'll discuss about that detailed functions of endoplasmic reticulum and Golgi complex vacuoles. Right? I'll discuss now. Yeah, this is one of the endomembrane system. Within that, the very first part is here, endoplasmic reticulum. This is one of the endoplasmic reticulum. See, 7th class onwards we are studying about endoplasmic reticulum. See, there are two types of endoplasmic reticulums are there: rough endoplasmic reticulum, smooth endoplasmic reticulum. See, both these are included in that endomembrane system, either rough or smooth, both are, right? So here, this is one of that endoplasmic reticulum which has that ribosomes. The endoplasmic reticulum, especially a rough endoplasmic reticulum, have the ribosomes. So presence of these ribosomes, right? The protein synthesis will take place. After that protein synthesis process, the proteins are transported. To Golgi complex. The endoplasmic reticulum, especially this is RER, rough endoplasmic reticulum. That rough endoplasmic reticulum involving protein synthesis. These are transported to Golgi complex. Here, after that protein synthesis process, the proteins are entered into the Golgi complex, right? So, why the proteins are entered into the Golgi complex? Whatever that proteins are synthesized by RER, these are inactive proteins. The inactive proteins by the Golgi complex, the inactive proteins are converted into the active proteins. These proteins are active. So, what I am telling here, that Golgi complex is responsible for the activation of proteins. So, the step is called as post-translational modifications. By the endoplasmic reticulum, especially a rough endoplasmic reticulum. By the endoplasmic reticulum, translation takes place. Translation is nothing but synthesis of proteins. After that translation process, by the Golgi complex, activation of proteins takes place. The step is called as post-translational modifications. Post-translational modifications. So the difference between these two, after the translation, here after indicates here post. After the translation, that one is indicates here post. So after the translation process, post-translational modifications takes place. After the translation process, post-translational modifications takes place, right? So that is about the Golgi complex functions. So how this takes place? For suppose this is the endoplasmic reticulum, this is one of the protein, and this is one of the vesicles-like structure. And this position, especially within that Golgi complex, there is two positions are there. One of the position is called as cis position, and this position is called as trans position. So whatever that inactive state proteins are synthesized by rough endoplasmic reticulum, these proteins are transported and fused. Then the inactive proteins are entered into the Golgi complex. The inactive proteins, some of the modifications takes place within the Golgi complex. To the inactive proteins, some of the modifications takes place. By that modifications, the inactive proteins are converted into the active proteins. Simple. Active proteins. Simple. Here cis position is there. At cis position, inactive proteins are fused. At trans position, whatever that vesicles like structures are there, these vesicles contains the mature proteins or active proteins. So here within that Golgi complex, there is a long tube-like structures are there, or tubules-like structures are there, and vesicles-like structures are there. Right? That Golgi complex is responsible for the activation of proteins. Means by the deletion process, the inactive proteins are converted into the active proteins. So here this is about that cis and trans. At trans positions, there is a vesicles-like structures are formed, and the vesicles-like structures contains the active proteins. Sir, why here vesicles? Here vesicles are nothing but vacuoles or lysosomes. Okay, these are also single membrane structures. This one is single membrane structure. And these are also single membrane structures. So the vacuoles are the lysosomes which contains that active state of the proteins, and these proteins are transported from one location to the another location, means inside the cell. The synthesized proteins are transported from one location to the another location. So whatever that proteins are synthesized, and these proteins are activated, and these are packed. After that packing process, these are transported. So vacuoles and lysosomes, which are supporting for the transportation of active proteins or enzymes. So that is about the endomembrane system. So what I am telling here, remember all of you, within endomembrane system, three parts are there. One is endoplasmic reticulum, involving, especially rough endoplasmic reticulum, involving protein synthesis. Once the proteins are synthesized, majority of the proteins are inactive proteins, and these are activated by Golgi complex. This step is called as post-translational modifications. After that, activation process, these are packed. After that packing process, which are transported within plant cells by the vacuoles. Within animal cells, these are transported by lysosomes. Remember, the transportation not only inside the cell, the transportation of enzymes, the transportation of proteins, not only inside the cell. Sometimes extracellular secretions also takes place. Extracellular secretion means some of the proteins transported from inside the cell to outside the cell. Right? So that is about the endomembrane system. Right?